Hello, my name is Santiago Alvarez, and today I'm going to expose my seminary, which is about using restriction endonuclease protection, selection, and amplification to identify preferred DNA binding sequences of microbial transcription factors. General objective. The general objective is to expose the use of restriction endonuclease protection, selection, and amplification to identify preferred DNA binding sequences of microbial transcription factors. In introduction, first I want to talk about microbial transcription factors. They are proteins capable of specifically binding to short DNA sequences located in gene promoters, and these which activate or repress transcription target genes typically in response to an environment or cellular trigger. Also, it's important to say that they work as regulators of transcription. Another very important thing to mention is the restriction enzymes. A restriction enzyme that also has the name of restriction endonuclease is an endonuclease that can recognize a characteristic sequence of nucleotides within a DNA molecule and also can code this DNA in that specific point. Also, other thing very important is that different restriction enzymes recognize and code different DNA sequences, like depending of the investigation that is going to carry on. In the method, first it's very important to talk about the RIPSA. There is restriction, endonuclease protection, selection, and amplification. This RIPSA consists of several rounds in which a set of DNA is incubated with a transcription factor. This transcription factor is of interest and is challenged with an 2SRE. Like this, 2SRE is the meaning of type 2S restriction enzymes. The first round of RIPSA utilizes a selection library of DNA, contemplates containing a region of random nuclear sequences, and also it's very important to say that each round selects for prepared DNA binding sequences, which are then subjected to a PCR. And also this PCR is used as inputs for the subsequent round of RIPSA. Other important thing is the DNA purification. This will depends specifically in the tissue or cell samples that you're going to use. Depending on this, you can use chemical treatment, enzymatic digestion, or mechanical disruption to elicit the DNA. And in this order of ideas, you also can remove those contaminants from the nucleic acids. And what is the purpose of this? This will make more convenient for storage and handling these samples for the investigation, and in this case it will be very important to find those binding sequences of DNA.
Also in methods we have the PCR. This is a laboratory technique for roughly producing millions to billions of copies of a specific segment of DNA, which can be used for a greater detail. Also, PCR enables the synthesis of specific DNA fragments using a DNA polymerase enzyme, which takes part in the replication of the cellular genetic material. Also, we also have the restriction enzymes. In this, as we said before, these have the function of recognize a specific sequence of DNA and also create there. The one that they use the type 2S restriction enzyme, and also this will help to analyze it in a better way. Bueno, entonces aquí en los resultados lo que podemos hallar es que tenemos las tres muestras de ADN, en las cuales unas no estaban cortadas, otras estaban cortadas con esta enzima de restricción, la 2S, y otras tenían la enzima de restricción, pero también tenían el factor de transcripción, el RAR. Entonces, lo que podemos analizar con los resultados es que en los primeros ciclos son casi muy parecidos los pares de bases, o sea, es muy parecido entre los que están cortados y los que tienen el factor de transcripción, pero cuando vemos a muchos más ciclos, podemos ver que obviamente aumenta la población del ADN, y de esa manera también se puede evidenciar que el REPSA salió exitoso. ¿Por qué? Porque se ve que hay presencia de DNA que fue protegido por ese TNF frente al corte de la enzima de restricción. Entonces se puede decir que ese REPSA fue exitoso, y en el artículo mismo lo dicen, que llegó a que es un 70%, que es muchísimo más de lo que esperaban, y se puede mostrar esa evidencia de esa unión del factor de transcripción a eso, protegiendo el corte. Bueno, aquí en los resultados podemos seguir viendo como esa eficacia del REPSA de que realmente está mostrando de que de cuando se aumentan, bueno, cuando se someten a esos ciclos de PCR, en especial cuando ya están en más rondas más avanzadas, se logra ver como esa secuencia que con el factor de transcripción se logra mantener como unida y por ende se nota que la banda está más arriba, generando que tiene mayor como concentración de pares de bases. Bueno, y esto se puede representar tanto en la una y en la de la derecha. Podemos ver más como un esquema de cuando se someten como a esos ciclos, ¿cierto? Y en los esquemitas que podemos ver a la derecha, se refiere más a cómo sería como esa secuencia, como esa organización de nucleótidos dependiendo del número de pares de bases de donde quedó registrada la banda. So in the discussion, I choose three authors. The first one is Wang. He said that for the mechanism of action of a folk, like for cleavage, they have to be two monomers, and we agree with that one. Also, next one, there is Hardenball. He said that REPSA also function like with other ligands, for example, as in nucleics, and we agree with that one also. And the third one is Banshibal Golpachin, and he said like also REPSA works like with other non-ligands, not protein ligands or non-transcription ligands, and this also we agree with that one. So, in conclusions, the first one is that REPSA technique is in fact a functional technique to identify those DNA binding sequences, and with them we can continue investigating and deepening about the transcription regulatory network. All of this based in embryological function. The second one conclusion is that in medicine this is very important because many diseases have a relation with this regulation of transcription factors, like cancer. So with this technique we can be more precise with those diseases and also with the therapeutic solution for them. Thank you.